acetyl-CoA will join up with a 4-carbon molecule as it enters into the TCA cycle, so that we generate now a 6-carbon molecule. The CoA goes away. So now we have a 6-carbon molecule here. That is entering into the tricarboxylic acid cycle. Again, you might notice the Krebs cycle.
Citric acid is carbon at 6 carbon.
I'm sorry? So yeah, this is a citrate molecule. That's just what it's called. It's a citric acid molecule. We're starting the Krebs cycle. They don't call it that anymore. They call it the tricarboxylic acid cycle. You can still call it the Krebs cycle if you want. So just so we're clear on what we've done so far, we've taken 6-carbon molecules, broken it into 2 3-carbon molecules, taken those 2 3-carbon molecules, and each of them have been broken into 2-carbon molecules, which have then been added into this TCA cycle. Now, what we're going to see happen is, again, through a series of enzymatic reactions, we're going to break down this molecule. We're going to release a CO2 molecule through one of these reactions and generate some NADH. So some more reducing power. Here's a 5-carbon molecule now. Could serve as a precursor metabolite. Does serve as a precursor metabolite, meaning that this molecule could enter this cycle here, or it could be siphoned off if it needed to for something else, depending on what the cell's needs might be. Again, we're going to break this off, release some CO2, release some NADH, and now we're at a 4-carbon molecule. We've completed the breakdown of one of our pyruvate molecules, haven't we? If it was 3-carbon when it started here, and we've come up, broke down, or released the CO2 here, so we broke it down to a 2-carbon molecule, then it entered where we added it to a 4-carbon, released another one, and released another one. We've essentially released 3 carbon molecules, haven't we? We've essentially released one or completely broken down one pyruvate molecule. But we still have the other one. So the cycle has got to continue one more turn to complete the breakdown of the other pyruvate molecule.
What's PCM?
Precursor metabolite. So pyruvate is a precursor metabolite. It's just that metabolic intermediate. Now, as we move around this cycle, we're going to generate some ATP here. We're going to generate some FADH over here. This molecule here is a precursor metabolite. I wouldn't really care that you know that. Just another molecule in the process here. One more NADH molecule here. And we've completed the cycle for the tricarboxylic acid cycle. We're generating 1, 2, 3 molecules of NADPH and a molecule of FAD with each turn of the cycle. And we have to recognize it's going to turn twice for every pyruvate molecule that we have.
So all those up there are molecules.
So we've generated 1, 2, 3, 4 NADH, that's what you said?
Here we've got 3 with the turn of the cycle. Each time the cycle, and 1 FADH. Well, we've got an NADH over here, and we've got an NADH over here. What was the question?
All those are molecules up there?
Yes. So if we were to count this up, what do we get from our TCA cycle? We get 2 ATP, don't we? We got 2 ATP, 1 for each turn. We've got 6 NADH molecules. We've got 2 FADH molecules. And so we're done, aren't we? Not really. We've finished the breakdown of the glucose molecule, but look at, we've got this molecule that's high in energy, and actually there's 2 of them here because of the fact that that's happened twice. We've got this energy high in reducing energy, or this molecule high in reducing energy, and we actually have 2 of those. We've got all of these that we've just generated, high in energy. A couple of things I want to point out before I move into the next step. This process here, the tricarboxylic acid cycle, is happening inside the matrix of the mitochondria. So if this is our mitochondria, this is the inner membrane. The tricarboxylic acid cycle is happening right here inside the mitochondria, inside the inner membrane. Pyruvate has to move into the mitochondria in order for TCA to continue, in order for the continued breakdown of the molecule. So that brings us to respiration. Respiration is the final step in the metabolic pathway here in the generation of energy. It's going to use those molecules of reducing power to generate ATP by oxidative phosphorylation. And it's going to employ the electron transport chain. Imagine okay, so I know my tails didn't go long enough, but this is our membrane now, okay? If we were to look at our mitochondria again, going up and looking at our mitochondria, I'm taking a section right here from the membrane, the inner membrane of the mitochondria. What we find on this inner membrane are a number of proteins that are embedded in this membrane that serve to transport that electron from one protein to another. This is complex 1, this is complex 2, this is complex 3, this is complex 4, and then there's also a couple of molecules that move between these. So these molecules are pretty stationary, embedded into the membrane. These molecules here move freely through the membrane. They're all proteins. Okay. So, I've got my NADH molecules, right? I've produced these NADH molecules inside the lumen of my mitochondria. This NADH molecule comes up here and transfers the 2 electrons that it's carrying to the embedded protein, if you will. And what this does in transferring these high energy electrons is it allows for the protein then to move out of the cell some hydrogen ions. So we are now moving hydrogen ions out of the cell with the transfer of these high energy electrons to complex 1. Now what happens is that this protein will pick up those high energy electrons and transfer them over to this complex here. And in doing so, we will pump more hydrogen ions out of the cell. The transfer of electrons continues with this molecule picking them up here and moving them over to this complex of proteins. Again, using the energy from those electrons to move protons out of the cell. I didn't use this one here. Reason for that is because this is where FAD comes into play. FADH will give up its electron to complex 2, where it can be picked up by this quinone and transferred to the other complexes. So FADH enters in a different place, and so it doesn't generate as much energy as NADH does in this chain. Now, if we think about what's happening to our electron, we can think of it as a stair-step decline in energy. If this is where the electron energy is when we start the cycle, over here with NADH giving it off to the complex 1 here, each step the electron loses some energy. So each time we transfer it to a different molecule, we're giving up a little bit of energy in order to transfer the hydrogen molecules outside the cell. So at the end, we end up with an electron that's been spent. The energy's gone. Complex 4 transfers that electron now to an oxygen molecule to form water. Okay?
So if it's losing electrons on each step, it comes in a complex.
It's not losing electrons. The electron is losing energy.
Energy, okay. So does that change the gradient of how much hydrogen's going out? Like at complex 1, you have some, but you've lost some energy by the time it gets to complex 3, so you're pumping out less hydrogen each time?
Well,
the electron
that depends on the complex. How much is getting pumped out is all dependent on the complex, not the energy in the electron. Because they're using the same amount of energy each time. Okay, imagine that the electron's energy, this is the electron's energy. Okay, so the first time they use this much energy. The second time they use this much energy. So they're using the same amount of energy for the most part each time they carry out a reaction. Not necessarily every single time, but you can see, so there's not really a decrease of energy used to pump the ions out. They're still using the same amount of energy. It's just that the energy in the electron is going down now. So if they use 50 watts, if your electron had 200 watts, or I don't know if that's right, I'm not an electrician, but if they used 50 units of power here, say your electron has 200 units of power, they use 50 units here, they're going to use 50 units here, they're going to use 50 units here. They're going to use the same amount of energy, for the most part, for each reaction. But at the end, after using 50 units each time, you're down to 0 units. So that's what's happening here. Your electron is spent by the time it gets here. No energy left, and so they give it off to the oxygen carrier.
So then it doesn't change how it functions, it just gets used up.
It just gets used up.
Okay.
Yes.
The function, it works the same at the same level until it's used up.
Yes.
Okay.
Yes. Okay, so now we've established this proton gradient. Well, we haven't generated any energy, have we? No, we haven't generated any ATP. There is another protein that is embedded in the membrane called ATP synthase. ATP synthase now allows the hydrogen ions to flow back into the cell, serves as a gate, opens up, allows the hydrogen to flow back into the cell. And this doesn't require energy, right? Because we've got lots of hydrogen ions up here, and we have very few here. So the gradient is such that the ions are going to want to flow this direction. We've used the energy of the electrons to establish this gradient so that those hydrogen ions will flow this direction. As they flow this direction, you can think of it as like a water wheel. You know, as water flows under the wheel, it turns the wheel, and as you turn the wheel, you generate electricity. Same thing here. As we turn that wheel,